Thank you all for coming. For those of you who are here in the auditorium and for those of you who are following the event through Twitter. I'm Maureen Goodenow. I'm the Director of the Office of AIDS Research here at the NIH, and I want to thank you again for joining us in observing World AIDS Day 2018. As most of you are aware, World AIDS Day actually started December 1st, 1988, and it's a day for us to unite in the fight against HIV, to support the approximately 37 million people worldwide and the 1.1 million people in the United States with HIV, and to remember the 35 million individuals who have died from an AIDS-related illness since the beginning of the pandemic. OAR is delighted at today's turnout from across the NIH, HHS, and the community for this event, which is titled More Than 3 Decades, Inspiring HIV Discoveries Through Basic Science Research. In keeping with the federal theme of this year's observance, Saving Lives Through Leadership and Partnerships, this morning you will hear researchers from NIH and from leaders and partners in the HIV field who will discuss the role basic science research plays in the ongoing fight against HIV. Conducting basic science research is a major part of the NIH mission. NIH Director Dr. Francis Collins wrote about the importance of this research in a blog post earlier this year. Basic research generates fundamental knowledge about the nature and behavior of living systems, and this research promotes the biomedical advances needed to protect and improve our health and the health of generations to come. The ultimate goal of this research is to end the HIV pandemic and improve the health of people with HIV. OAR coordinates the NIH HIV research agenda. As the HIV pandemic crosses nearly every area of clinical medicine and basic science investigation, the NIH response requires a unique and multidisciplinary global research program that transcends nearly every institute and center. And currently, as illustrated on this slide, 23 institutes and centers and 3 offices across the NIH conduct HIV research. NIH basic science research provides the foundation for all HIV research areas. HIV research changed the basic research paradigm in academia. Also consider the impact HIV research has had on basic science infrastructure, accelerated review process for applications, biorepositories, clinical trial networks, research collaborations through centers, program project grants, and novel mechanisms for funding. Basic research has led to remarkable scientific achievements that benefit people with HIV. In the 31 years since the first World AIDS Day, what was a fatal disease is now a chronic condition with treatment. Basic scientific research has played a key role in developing effective treatment and prevention tools to bring about this progress. The basic science really sets the course for discoveries in a pipeline that leads to public health. And what we're going to be focusing on today is very much at the beginning part of this pipeline in basic research, but we're also going to be hearing talks about translation and clinical applications and studies that are going to be leading, hopefully, for development of a vaccine and real impact on public health. Basic research leads to improved health, and together with community stakeholders, advocates, and scientists within and without NIH, outside NIH, we have helped to charter the course that has led to many discoveries and breakthroughs and changed the face of the pandemic. Safe and effective interventions to prevent acquiring and spreading HIV have been developed, including PrEP, TASP, and now with antiretroviral therapy enables people with HIV to achieve undetectable viral levels, has led to the U equals U movement, or undetectable equals untransmittable, meaning people with HIV and their partners can be in a relationship without fear of transmitting HIV. This development is a game changer and helps to reduce stigma as well. So looking ahead, While basic research has led to significant scientific achievements, challenges remain. How the virus remains hidden and becomes activated even with antiretroviral treatment. How co-infections and other health conditions develop and progress in people with HIV. We need an effective vaccine and we need a cure. So today's event, which is going to be a fantastic group of presentations that really span from basic through the clinical translational, and then we'll hear from our Library of Medicine about the data that's being generated, not only from the HIV field, but from other fields as well. Our event today is being moderated by Dr. Carl Dieffenbach. Carl is the director of the Division of AIDS in the National Institute of Allergy and Infectious Diseases. He has been a really fantastic colleague before I joined the NIH and since that time. And he's really here today because I see him as the NIH raconteur par excellence. So I will now turn this over to Carl.
Thank you so much, Maureen. If it's okay with everybody, I'll just stay here. What I'm going to do is moderate today's session. I will introduce each speaker individually and then come back at the end and we'll have a roundtable with questions that I have prepared to pepper the panel with, as well as hopefully questions from the audience. As Maureen has said, basic science is the understanding of the mechanisms of specific biological events. And as we think about the kinds of the biological events that we study that have led to essential public health changes, something as simple as what it takes to incorporate a nucleoside base into a replicating nucleic acid and how we target that with antiretrovirals that essentially interrupt that process is extremely important to understand. And our ability to make those drugs grew out of our understanding of what it took to make a nucleoside that could be differentiated between a reverse transcriptase and a human DNA polymerase. Additionally, the kinds of things that are needed for vaccines are an understanding at the molecular level of what it takes for an antigen to trigger a B-cell receptor. These bricks of basic science make up the blocks and foundation upon which we create vaccines and medicines to prevent and treat diseases. We have 7 speakers today on topics that relate from everything through drug discovery, vaccines, through big data. So if you'll allow me, we'll move on and I'll introduce the first speaker. It's my pleasure to introduce my colleague and friend, Dr. Larry Corey. Larry is the President and Director Emeritus of the Fred Hutchinson Cancer Research Center and Professor of Medicine and Laboratory Medicine at the University of Washington. He's also Principal Investigator of the HIV Vaccine Trials Network. You have a fairly extensive biography of him in your notes. I will note that Larry is a University of Michigan grad for both medical school and his undergrad. The title of Larry's talk today is The Road Less Traveled, Perspectives on an Effective HIV Vaccine. Larry.
All right, Carl, thank you. And Maureen, thank you for inviting me here. Thanks for the kind introduction. So I'm going to, as the first speaker, I just will sort of introduce a little bit about the issue of, why are we talking about HIV and why do we need an HIV vaccine? Because it really is the epidemic of our generation, and still is. People living with AIDS, these are mind-numbing figures, are 37 million. New HIV infections in 2017, 1.8 million, and still over essentially 1 million AIDS deaths. That translates to about 5,000 new HIV infections every day, even in 2017. Yes, 2/3 are in sub-Saharan Africa, 500 a day or 10% are among children, 4,400 among adults ages 50 years and older, and 43% upon women. So, and while we have started to bend the curve, so to speak, when it comes to the issue of transmission, if we look at this, line, yes, we've started to look at this, but if you really look at the issue of where we are at 1.7, 1.8 million, and you follow the trajectory here, we're still talking about 1 million new infections out to 2040. So bending the curve is something that we actually need to do well. And while there may be sort of press boredom with HIV, the virus is thriving, and it's for us scientifically to look at this. Obviously, deaths are still occurring, and maybe this is doing better, but access to therapy and keeping people on therapy is still a very important part of our field. We in the United States really sort of just are, I'll say the word, tolerating an epidemic of 40,000 new infections, which have not bent or changed over the last decade. So 40,000 new infections. We hear all sorts of things about 1,000 cases of Ebola. I'm not underestimating that. But again, we seem to have this sort of tolerance over a long period of time of these chronic 40,000 new infections in the US. About half of them are in the South. And epidemiologically, they are concentrated. Over 50% of them are in 46 counties throughout our country. And some concentrated area of both prevention, research, and implementation through what we learned in smallpox, ring epidemiology and ring science should be thought about as it relates to our country and where our epidemic is. Outbreaks are associated with the opioid epidemic, and we do get outbreaks. This is an outbreak associated with an opioid epidemic in rural Ohio that had over 157 cases in a rural county over a short period of time. Frankly, with asymptomatic acquisition, prolonged subclinical infection, and sexual transmission, getting to an AIDS-free generation will require a biologically based primary prevention modality with prolonged durability. Traditionally, this has been an HIV vaccine, whether we approach this with a long-acting antiretroviral, or I'll tell you about long-acting broadly neutralizing antibodies. But to me, an AIDS generation is a 95% reduction in incident cases, which is for us less than 2,500 cases per year, which is not 40,000, and globally is less than 100,000 per cases per year. And this will require concentrated both scientific and policy issues. Now, it has been hard to develop an HIV vaccine. Lee Hartwell, who preceded me as president of the Hutch, asked me when I took over the HIV vaccine program or started it, did I think I would be successful? And I said, sure, give me a decade. Well, we're at 18 years later. But we're persisting, and we're making really large inroads. The genetic diversity of the virus is greater than any other pathogen. The envelope is less immunogenic. We really don't know why. We can take any vector, put a different kind of protein in, Zika, Ebola, rabies, and we get an effective vaccine. But we take that same vector and put an HIV envelope, and we get a lot of the work that we've done in the HVTN has actually led to the Zika vaccines and the Ebola vaccines, but the same technologies are actually not working as well for HIV. The trimer is unique. It's hard to simulate. There are a few trimers on the surface. It's the baldest virus that we know of. And the immune response actually seems to slip off, and you don't get bispecific antibodies and antibodies that adhere with high binding affinity. The animal models are expensive and at the moment non-predictive of vaccine efficacy. And there are no human cures of HIV, and hence there are no models to mimic. And we're sort of 0 out of 65, so of 65 million. So we actually need to be cleverer than we have than natural infection. We can't just mimic natural infection. Now, the good news about the status of HIV vaccines is there is a robust pipeline, especially new immunogens that are dedicated to eliciting neutralizing antibodies to the virus. I'm not going to talk much about that because our major progress at the moment is in these prime boost regimens that elicit non-neutralizing antibodies that we can now have enhanced magnitude as compared to the 1 partially effective vaccine that was seen in RV144. And I'm going to tell you about these vaccines that equal the immune responses among non-human primates who are protected in mucosal challenge studies with HIV, as well as the human studies that have been done with these concepts. Really, from an HIV vaccine point of view, we have 3 efficacy trials, the first time we've ever been able to do that in the field, ongoing. One of it is called the Antibody-Mediated Protection Study, the other HVTN-702, and the other one HVTN-705, and I will talk about them quickly. One of them is essentially built upon the RV144 trial, which was a recombinant canary pox virus with a GP120 boost that most people have heard it had a trial of approximately 30% effectiveness. What happened after that was really a wonderful collaboration internationally of scientists to look at the correlates of protection. What were factors among the people, the 31% who were protected versus not? And it was shown that it wasn't neutralizing antibodies, but antibodies to part of the virus that we actually didn't spend enough attention to. Actually, in many early vaccines, that's this part of the virus that was actually clipped out. Antibodies to this V1, V2 region were associated with a reduced risk of infection. Monoclonal antibodies taken out of the serum for people who got this vaccine showed that they had ADCC activity rather than neutralizing antibody. And there's been a considerable amount of work on how to prevent infection and how to make immunogens that would improve upon this. When we look at the transmission bottleneck that came out of our RV144, they were non-neutralizing. They killed infected cells through ADCC and ADCP activity, and they eradicated the initial nidus of infection. This is just correlative risk that really showed that the higher the magnitude of that immune response, the V1, V2, the better the efficacy one had. And one could also show that it put pressure on the virus at the point of attack where these antibodies were directed. A partnership was formed between the NIH, and the Bill and Melinda Gates Foundation, and the pharmaceutical industry and the U.S. military research program to sort of build upon this and construct a series of vaccines that would improve upon this. It was called the P5 or Pox Protein Partnership. And this led to the development of an all-vac-based vaccine associated with 2 GP120s that were Clade C directed, and that we would go into the sub-Saharan and Clade C population and see if we could build a vaccine that had better immunogenicity than RV144, better durability, and that we would then take into efficacy. We did a qualifying trial to build these vaccines. Qualifying trials showed that it had excellent immunogenicity. And in November 1st of 2016, we started a 2B3 trial that was to take heterosexual men and women in sub-Saharan Africa. It's a 5,400 person trial. As of today, about 4,700 of those 5,400 have been enrolled into the trial. It's a regimen that, yes, it's a complicated regimen. We've had to give boosts at month 12 and month 18. We're looking at 3-year efficacy. Again, durability is an issue, and it goes with all of HIV vaccines, but the issue is to get prolonged efficacy in order to make an effect. And this trial is ongoing. Like I say, it's about halfway through. It's 3/4 of its enrollment, and we have about another 18 months before we really hit a serious mark with respect to the DSMB. There is another regimen that is also based on neutralizing antibodies. It is initially developed through work at the NIH with a recombinant Ad26 virus that was supported through Beth Israel and Dan Baruch through the various grant mechanisms that NIAID have for vaccine development. It has then commercially been picked up by Janssen and J&J. And that is a trial that is called HVTN-705. And that is based on a concept that came essentially out of one of the trials in HIV that failed, the STEP trial. That was a vaccine trial that was led by a vaccine that was developed by Merck, in which we actually showed that even though you develop T-cell responses to GAG, POL, and NEF, those T-cell responses were immunodominant, and that did not cover the diversity of the virus. Out of that came the concept that how do you get better coverage? And you get better coverage by designing, using synthetic biology to design vaccines in which the immunogen covers the strains that were developed. And Betty Korber came up with an idea of developing what we call mosaic vaccines or synthetic mosaic vaccines. And this is really using synthetic biology at its highest. And the antigens that are used in this vaccine are mosaics. It's a complex vaccine that has mosaics of two envelopes, two mosaics of envelopes, which cover essentially all the clades as well as mosaics of the GAG, POL, and F. So it's a 4 mosaic vaccine and an Ad26 vector, sort of 4 separate mixtures of the Ad26. And it has the component of, again, a heterologous protein boost. This time a GP140, I can't really say it's a trimeric structure, but it is a trimer that is a clade C trimer that is given in this regimen of 0, 3, 6, and 12. This vaccine has been highly effective in the non-human primate mucosal challenge, and it's approached in a slightly different way. The concept was to look at the animals that were protected, look at their immune responses, and ask the question, what are the correlates in those animals? And again, they were non-neutralizing antibodies, were the correlates ADCP and high GP120s. And then to take these immune responses between the protected animals and the unprotected animals, come up with the criteria, and then ask the question, can we build a vaccine in humans that actually meet this criteria? And do this in phase 1 and phase 2. And these are the data. These are the human data from what there's a phase 2 study that was called the APPROACH study. And these are the non-human primate data. Humans actually exceed it. And that's the basis of taking this vaccine into an efficacy trial. And this is, again, a randomized placebo-controlled trial looking at this vaccine in women in sub-Saharan Africa. It's classic 1-to-1 randomization of 2,700 women at high risk of HIV infection. That trial started about a year after 702. Of the 2,700, we currently have 1,200 enrolled. And this is being done in South Africa as well as Botswana, Zimbabwe, and Zambia, as well as Malawi. So that trial is ongoing and, again, is doing very well. And we're probably 18 months to 24 months from hearing anything about that. So we have 2 non-neutralizing strategies that are being undertaken. One is based upon RV144 and the human correlates. One is based on the non-human primate challenge experiments. Both approaches suggest correlates relate to both binding and functional antibodies, ADCP and ADCC, as well as, I haven't really gone over it, but there are T-cell responses, CD4 envelope responses associated with this, and it's actually the T-cells that seem to make CD40 ligand, which is important in the T-cell B-cell interaction. So we do feel this is more of an antibody mediated, but it's the crosstalk between T-cells and B-cells that are important here. We shall see whether these presumed correlates are shown to be consistent in human efficacy trials. We shall see if the non-human primate challenge studies are predictive of vaccine efficacy. I truly hope so. In the end, we may have to take both neutralizing and non-neutralizing antibodies to achieve success. How much we can use and push non-neutralizing antibodies to make a vaccine is what this program is all about. Now, I do want to shift to neutralizing antibodies, which is sort of the virologist's classic approach to prevention. The last decade has brought the recognition of cloning B cells and recognizing where neutralizing antibodies occur to HIV. There are a number of areas where those neutralizations have occurred. And about 2 years ago, working with Carl and with the Vaccine Research Center, we started the concept of taking one of the neutralizing antibodies that was developed called VRC01, Vaccine Research Antibody 01, which binds to the CD4 region, and putting that in what I think is one of the great clinical tour de forces ever done. Of taking this neutralizing antibody, giving it intravenously every 8 weeks to 2 cohorts of people, 2,400 MSM and transgender men and women in North and South America, and 1,900 women in sub-Saharan Africa. This is a trial that is fully enrolled. So that's 4,600 people. They get 10 infusions. So that's 46,000 IV infusions. We've passed 35,000 IV infusions. We do not have one infection at the site. These are amazingly safe antibodies. We are fully enrolled. There is 95% retention and 99% adherence. So if there is altruism in this world, it exists. This is an amazing clinical trial, both with respect to the logistics associated with this, the clinical trial sites throughout sub-Saharan Africa, Peru, and the United States that are doing this trial with this kind of adherence and this kind of retention. Why are we doing this? We're doing it to find out what's the level of neutralization. We already know we can improve upon VRC01. We have way more potent antibodies. We know we can give these antibodies subcutaneously, and we can give them every 4 to 6 months. So this is a concept study. This is a study to tell us what's the level we need to do. And then we really know that we already have better antibodies that we can put together into a cocktail that we can give subcutaneously in a very practical way. We just need to know what the level is, and then we move into really defining what is a clinically relevant study. So it's an exciting time to be in vaccine discovery. The science is advancing through clinical trials. There are 3 pivotal HIV vaccine-related efficacy trials are underway, AMP, Alhambra, and Imbocoda. These trials will define if either or both neutralizing and or non-neutralizing antibodies can be tweaked to provide reasonable vaccine efficacy in high-risk regions of the world. Antibody isolation and characterization has revolutionized our understanding of the immune response. Technological advances allow researchers to understand where antibodies target the virus in unprecedented detail. Stabilization of the HIV envelope trimer allows for engineering of trimeric mimics. I didn't talk about that, but that is where the field is going. And we have shifted essentially from empiric approaches to hypothesis-driven approaches. And that is both gratifying scientifically and much more likely to succeed. The next generation vaccines are entering the clinic. These are native-like trimers meant to resemble HIV envelope spikes. I didn't have time to talk about this. And germline targeting approaches that are generated using structure-based vaccine design. So the next World AIDS Day, I hope to be able to tell you more about that, as well as where this marked progress is. So it's with, I guess I'll say, cautious optimism that we are in this field of HIV prevention. It's a pleasure to be here today, and thank you.
Thank you, Larry. Moving on, our next speaker is Dr. Avindra Nath, Senior Investigator at the National Institutes of Neurological Disease and Stroke. Avi joined NIH as the Clinical Director of NINDS in 2011. And is also the director of the Translational Neuroscience Center and chief of the section of infections of the nervous system. Dr. Nath's research focuses on the understanding of the pathophysiology of retrovirus infections of the nervous system and the development of new diagnostic and therapeutic approaches for these diseases. This morning, Dr. Nath will discuss HIV reservoirs in the brain, advances in detection and elimination. Avi.
Yeah, thank you very much, Carl, for that nice introduction. So I'm going to tell you a little bit about what's happening with HIV in the brain and where the current state of the art is in detection and with our approaches to elimination of the virus. So I've divided my talk into 4 parts. The first is, where is virus in the brain? As you know, that the virus largely resides in T cells compartments within the rest of the body, but there's no T cell compartment within the brain. So the virus has to adapt and exist in other cell types. Then I'm going to talk to you a little bit about what happens to the HIV reservoir with prolonged antiretroviral therapy, and then our approaches to silencing and eliminating it. So the virus is distributed in certain parts of the brain, not all over uniformly. So within the meninges, you have a lot of meningeal macrophages. And if you look at these meningeal macrophages, there's ample amounts of virus there. You find them at the blood-brain barrier. And if you look at the brain itself, most of it is sitting around the basal ganglia and the frontal cortex and somewhat in the temporal regions, while the posterior parts of the brain are relatively spared. So if you look within the brain itself, you'll find multinuclear giant cells, you'll find perivascular macrophages. There's a lot of virus in the perivascular regions. And those can be detected by immunostaining. Then there are astrocytes. In some patients, they will get infected. They're harder to recognize, but you can use RNA or DNA hybridization to find these infected cells. You can also find HIV protein here, TAT aggregates sitting extracellularly. So these will form large aggregates and will immunostain here in a microglial nodule. And here in perivascular cells, again, some TAT in a microglial nodule here. So an interesting thing is that while we know a lot about how T cells and macrophages get infected, we know little about astrocytes. So if you take infected astrocytes and infected lymphocytes and put them on astrocytes, they'll stick like glue. And here on the scanning EM photomicrographs, you can see the lymphocyte here, and this is the astrocyte at the bottom. And there are all these processes that come out from these cells, and they just wrap around the infected cell. And if you look at the junction here, these red dots are all the HIV particles, and you can see that at the junction, there's a lot of virus that gets shed. And so there's a great opportunity for the virus to enter the astrocyte in the cell-to-cell contact manner. And here again, under transmission EM, you can see the virus here. They form these little synapses. And within these synapses, the virus will interact with both cell membranes at the same time. And I think this is critically important for the virus to be able to enter. Once the virus matures, it doesn't enter the astrocyte. Okay, so what happens to patients who are on prolonged antiretroviral therapy? The thing is, it used to be thought that if you take antiretrovirals for decades, your cells will turn over and you really won't have a reservoir. Unfortunately, that hasn't really happened. So we have long-term reservoirs. And if you look at this cohort of patients we follow here at NIH, so they have been on antiretroviral therapy with virus suppressed for a mean of 15 years or longer. About 2/3 of them, you can still measure TAT in their spinal fluid. And it's not small amounts. You can find high picogram to nanogram quantities of TAT. And once they're positive, you follow them along every 6 months or a year, and you'll still find it to be positive. You do find some fluctuations, but by and large, once positive, they're always positive. And we can also find them in exosomes. So if you take exosomes from the spinal fluid, and here by Western blot analysis, you can see TAT in these exosomes here. And in some of them, you can also find TAR, which is the RNA that binds to TAT. There's not always 100% correlation between TAT and TAR, but oftentimes you'll find the two of them together. And then the next thing that Dima and I are very interested in is trying to see if we can use techniques to detect the viral reservoir in live patients. And it's a challenging task, but nonetheless, this is a paper that was published from the Emory group and now in Louisiana. And they took an antibody, made it into a PET ligand, and showed that they can measure it in the rest of the body, but the brain is still not easy to penetrate with these things. So the next advancement would be to try and make probes that can enter the brain in order to recognize the reservoir. Okay, can we silence the reservoir? And so there are many different approaches, but one approach is, this is fine. So is to what we call is shock and block, okay? And so one can potentially design antisense molecules. I think the problem with designing antisense molecules is because they're so sequence specific, you really have to find a region that will take care of all strains and clades at the same time, and that's no easy task. But nonetheless, we found that at least 3 of these, which we call antisense 1, 2, and 3, that span the region of TAT and envelope, really have a decent efficacy in different conditions. So here, the AS2 seems to be really good, but they have variable amounts of effect in vitro to HIV replication. But the other interesting thing is that they not only block the transcripts, but they block all the proteins. So if you block TAT, which is one of the first proteins to be formed, and you block it right in the beginning, then no P24, there's no GP120 being formed from these cells. So you can block the entire HIV cascade. And the other interesting thing is that if you put them in PBMCs, and here we have a fluorescent tag on them, you can see that they're stable for here 16 days, but we can take them out to 2 months. As long as you can culture the cells, these things are very, very stable. And they degrade the transcripts as well as and prevent the proteins from being formed. So essentially, you can silence these cells completely for prolonged periods of time. So we've now been fortunate to be supported by the Office of AIDS Research to take this further into some cochleates and see if we can do animal studies and see if we can get it into the brain as well. Okay, what about elimination of the reservoir? And that's a more difficult task, but people have not been shy. There are many different ways that people are trying to approach it. One is an approach called kick and kill, and there are all these approaches whereby you may be able to reverse the latency, develop therapeutic vaccines, use combination of these two, or checkpoint inhibitors, and Larry just now talked about broadly neutralizing antibodies. The problem with some of these is that once you cause immune activation with these agents, the CNS can only handle a certain amount of inflammation. It's a very tight environment there, and so you'll end up with encephalitis. So one has to be very careful. Antibodies don't really penetrate the CNS very well, so that remains a challenge. What about checkpoint inhibitors? So checkpoint inhibitors are quite interesting because what happens with chronic antigen exposure is you get immune exhaustion. So if you can use an antibody to block these checkpoints, which here is PD-1 and PD-1 ligand, then you can reverse this immune exhaustion. And the cancer folks have done a phenomenal job showing that once you can do that, you can treat metastatic cancer and otherwise incurable cancer. So the HIV field is also very interested in trying to see if this approach can be used for eliminating the HIV reservoir. So there are multiple clinical studies ongoing right now, and we have one that I am a PI on, and here we're using a pembrolizumab, and our goal is to see what effect it has on the HIV reservoir. And these are individuals, this is the only trial in which we are using well-controlled asymptomatic HIV individuals. All these others around the country are individuals who also have HIV and malignancies at the same time. Okay, what about gene therapies? And so people can use genetic scissors these days using the CRISPR-Cas9 mechanism in order to either get rid of the receptor or the HIV genome itself. And how this works is you take these things, these guide RNAs will take the enzyme to the site, you cut it out, and then the DNA will repair itself. And so this is how gene therapy for any of these things would actually work. You can do it at hematopoietic stem cells, and then once you expand them, you can just transfuse them into these patients. And at least in animal studies, it's been shown that you can excise the HIV at various different places and actually cure a mouse. So that is certainly possible. Okay, so I'd like to stop over here and acknowledge the people here who've been doing all the work in my lab, and This is through collaborations. Sriram did all the scanning EM, and Farah did all the exosome work. Thank you very much.
Thank you, Avi. Our next speaker is Dan Appella. Dan is a senior investigator with the Intramural Research Program in the National Institutes of Diabetes, Digestive, and Kidney Diseases. Dr. Appella's research is really about synthetic organic molecules that are designed to to inhibit specific targets within the HIV replication cycle. This morning, Dr. Appella's presentation is going to discuss disrupting HIV maturation and infectivity with mercaptobenzimides. So take it away, Dan.
Well, thank you so much for the nice introduction. And thank you also to Dr. Goodenow for the kind invitation to come here and tell you a little bit about the research that I'm doing in my lab here at NIH. So I'm going to tell you a story about a particular molecule, which is shown on the front slide here. It belongs to a class of molecules called mercaptobenzimides. But the name of the character this morning, this particular molecule, I'm just going to refer to as 247. And it's a simple molecule by, I think, all standards. And compared to most HIV drugs, I would say it looks incredibly simplistic. Yet it has some really unique activities that could continue to fascinate me and hopefully will also fascinate you. I'm going to confess to you also, I do hope this molecule may have a chance at advancing to being helpful for treatment of HIV, perhaps prevention of HIV. And if not, this 247 molecule itself, perhaps a close derivative or analog that we're also working towards in my lab. And if that doesn't work, perhaps at least the basic science that we're learning from this molecule may help to identify new targets in which to attack HIV. Just a simple recap, certainly globally, this HIV infection remains a distinct problem with the burden of the disease largely borne by the poorer parts of the world. Many people receiving antiretroviral therapy, but still others not receiving therapy that is needed. It's remarkable we have 30 drugs now available to treat HIV infection used in combination, but still not a cure, requiring a lifelong treatment with antiretroviral therapy. I think it's fascinating also, there's a cost associated with this that is not always trivial. So about $20,000 to $36,000 per person per year in the United States. And after many years of therapy, what's becoming seen a little more obviously now is long-term toxic effects with continual medicines that are taken. Another fascinating aspect of HIV is the resistance that HIV develops to virtually any medicine. And one can see now in certain countries, hotspot areas, there's now 10% or more new infections that are resistant to frontline HIV therapy, prompting the need for perhaps new medicines and perhaps new directions for medicines to attack HIV. I'm going to go through a little bit of the activity for my favorite molecule, labeled 247. This molecule does have anti-HIV activity in cell-based assays. I'm showing here just 3 simple cell-based assays, as well as the activity listed by these EC50 values in micromolar. And as you can see from this, it's really not the most potent molecule. Certainly nothing in terms of potency compared to the more modern-day drugs. While it lacks potency, it has remarkable other features that I want to highlight to you today that I think are just completely fascinating. The first of all being on this column, where we try to look for the toxic concentration to kill cells, and we don't really find a toxic concentration. And this brings me to bullet point 1. This molecule is just remarkably non-toxic. Even in animal studies, where we've really pushed the consumption of this molecule in animals, we don't find toxic effects, such as animal death or other symptoms. The molecule also is active against wide varieties of different HIV viruses. We heard this morning in the first talk that HIV has a remarkable genetic diversity. And this molecule is remarkably consistent across a variety of different HIV clades shown here, all with the same micromolar potency. HIV develops resistance to known drugs. And again, our molecule has about the same antiviral activity against HIV viruses with known resistance to drugs. There are established drug targets within the virus life cycle. And our molecule doesn't seem to affect any of these established drug targets. And I'll talk more about this as I get along. But to start off, I'll just say that our molecule has no activity against HIV reverse transcriptase, integrase protease, or viral entry, which prompts the question about what exactly does this molecule do? This molecule can be used synergistically in combination. HIV is combination therapy, and we have examined the combination of our molecule with 23 FDA-approved drugs in cell-based model systems, and the molecule works nicely with those other known drugs, observing either synergistic or additive effects, and perhaps more importantly, no synergistic toxicity. Another aspect that really fascinates me is that while HIV is supposed to develop resistance to medicines, we are unable to generate resistant HIV virus in cell-based systems in the lab. And I'll admit here, after several attempts, we are just not able to find resistant HIV to this particular 247 molecule. I'll tell you, I am a chemist. My main concern is with making this molecule, and I can tell you this is not a difficult molecule to make, and it's rather cheap. In my lab, $15 a gram. We can make several grams, 10 grams, 20 grams in my lab, there's no problem. And we're working with chemical contractors to further improve the synthesis, and I can guarantee you this cost will come down as we get more experience with making it. What I want to highlight also is that our molecule does work as a microbicide, which I'm going to talk a little bit more about in detail today. So microbicides are prevention for HIV infection, specifically designed to prevent an HIV-positive individual from transmitting the virus to an HIV-negative person. It's a specific topical prevention to prevent people who have sex with others who are HIV infected. And there are 2 clinical trials that really bolstered the idea of a chemical intervention in form of a microbicide. One clinical trial called Caprissa showed that a 1% gel containing the drug tenofovir used by women in South Africa was successful at lowering transmission by a decent amount, 39%. This was in a gel formulation, which was applied right before sexual intercourse. Another formulation, which is perhaps more acceptable, would be to include the medicine into a contraceptive ring that could be used once a month. And a clinical trial called Aspire, using the molecule dipivirine, showed that monthly usages also were effective at reducing infections by about 30% in a clinical trial. Our molecule 247, at least in animals, does work as an effective microbicide. And I want to go through this study here, which is ongoing at NIH between my lab and the lab of Marjorie Robert-Guroff. Using rhesus macaque models as animals that are infected with the simian form of HIV, SIV, we have been able to formulate our molecule 247 as a 0.8% in hydroxyethyl cellulose, or HEC gel. Animals are challenged weekly for a total of 15 challenges, and we have 12 animals in the microbicide group, 10 in the control group. Challenge is performed 3 hours after application of microbicide, and overall the animals will be followed for 40 weeks after completion of the challenge, so the following is still ongoing. But I wanted to share at least the data we have so far, because the number of challenges has been completed, 15 challenges, and you can see that in the control group we now have all animals infected, whereas in the microbicide treated group we only have 2 of 12 animals infected, indicating that it is working as a microbicide. In any development of microbicide, it's important to also figure out, does this molecule have any kind of irritation? And again, in collaboration, this time with Ron Veazey at Tulane, using this molecule in gel with rhesus macaque animals shows really no obvious signs of inflammation. In these treated animals, histology of vaginal biopsies collected 7 days later shows no irritation with application and use of this molecule. Formulation of this into other formats, I mentioned the contraceptive ring format, is also important to advance the use of this molecule for compliance. And we have been successful here as well. This molecule 247 can be formulated into rings. We completed one study with Patrick Kaiser at Northwestern, formulating these into polyurethane rings. And we have an ongoing study now with Mark and John at the Oakcrest Institute of Science, where a ring is now drilled with these individual pods. Our molecule is incorporated in it, and we've shown that slow release of this molecule over 30 days is in consistent rate with what we would need for treatment. And the goal now is to get these rings into the hands of Ron Veazey to carry out these animal studies. So this molecule has some really interesting antiviral activity, but one of the basic questions we're also trying to get at is, how does this molecule work? I mentioned earlier, it doesn't seem to have activity against the known antiviral drug targets. So HIV is a retrovirus. It has RNA. We know so much about this virus and how it fuses to different receptors, the CD4 and usually CCR5, injects viral RNA integrates, and then eventually starts transcribing proteins with new viral RNA packages, and then goes off and matures to form a new infectious viral particle. The standard way of interfering with this virus are shown on these X's on this slide. There are entry inhibitors, reverse transcriptase inhibitors, integrase inhibitors, and finally the HIV protease inhibitors. And we're certain, after a lot of studies, that our molecule doesn't affect any of these traditional pathways. Instead, we believe our molecule is acting over here with the assembly process. And I want to go through at least a little bit of the evidence for this. First, I want to tell you at least a bit of the background for HIV, in particular highlighting on this GAG polyprotein. HIV, as it begins to transcribe itself, loves to produce a lot of this GAG polyprotein. And GAG consists of 3 important viral proteins linked together. It's HIV matrix, capsid, and nucleocapsid protein linked together by polypeptide chains. As GAG is expressed, multiple GAGs have to assemble around new viral HIV RNA through the action of nucleocapsid protein, shown in the blue dot, assembling around the new viral particle and driving forward the formation of what is initially an immature viral particle that then matures through the action of HIV protease, cleaving these bonds in the polyprotein of GAG to then take this immature particle and let the capsid core condense, where our HIV RNA is now nicely packaged. We believe we're interfering with this, and some of the best evidence we have comes from electron microscopy. In cells that are treated with our molecule 247, shown here as an HIV-infected cell treated with our molecule 247 with the resulting virus that comes off. And we repeatedly see by electron microscopy what looks like an immature virus particle locked in this immature state where the GAG polyprotein hasn't proceeded to be properly cleaved by HIV protease into what is a mature virus. As a control, without treatment, you can see an HIV-infected cell here now producing nicely mature HIV particles with a nicely condensed capsid core with the RNA in the middle. Going down further, we've conducted direct studies between our molecule and the HIV GAG polyprotein. Our molecule, shown here as 247, contains what we believe is a very unique kind of a reactivity to it, in which this portion of the molecule, the acetyl group attached to the sulfur atom, engages with GAG directly to transfer that group to very specific patterns along the GAG polyprotein. Shown here is a ribbon diagram of the GAG polyprotein. I've shown you the cartoon structure before with different domains of matrix capsid and nucleocapsid. Just to orient you, in this structure, the matrix of HIV is shown here. Capsid has 2 domains, the N-terminal and C-terminal domain, and the nucleocapsid portion is shown down here. We've studied the direct interaction of our molecule with HIV GAG, and we find a unique pattern of acetyl transfer, both to cysteines on the HIV GAG, followed by lysines on the HIV GAG. And what I've highlighted here, first by showing in the green, are the positions of lysines that get an acetyl group from our molecule. And you see that we're damaging GAG by acetyl transfer and affecting the lysines in green, as well as lysines that are purple on the N-terminal domain of capsid, more lysines down on the C-terminal domain, and a number of lysines on the nucleocapsid region. So at the end, what we're basically doing is modifying GAG in a way such that it looks like this cartoon, where now we have a variety of different acetyl groups on GAG protein, which we believe interfere with its eventual maturation of HIV. This is work that was done in vitro. What we have also seen is the ability to monitor this type of acetylation pattern and the impact it has on GAG in cell-based models, and we see consistent patterns. In this particular experiment, what we've done is treated HIV-infected cells with our molecule 247. This produces HIV virions, which we believe are immature, and we've taken these virions directly and tried to analyze them to look at the status of the GAG polyprotein in these HIV virions. In this gel, the Western blot, you can see in the first lane untreated examples showing fully processed GAG polyprotein. But if you look at HIV virions from treated cells and you look at GAG, it looks terrible. It looks like we've really done something bad to the GAG polyprotein with our molecule. This is a non-reducing gel. And under a reducing gel, this rather ugly looking band does sharpen up, as shown here, but again indicates a band on the Western blot showing that there is an unprocessed form of GAG that we have induced with our molecule. To further prove that it is our molecule responsible for this, we've carried out this experiment now using a radio-labeled version of our molecule where there's a C14 radionuclide on the acetyl group. And then doing the same experiment, we then find that the radio-label goes to the same band for unprocessed GAG polyprotein. So we think we have something very unique, and I'll say that we're still studying a lot of the aspects of this mechanism to try to uncover what it is about this molecule that can go in, modify GAG in such a way, and then result in a non-infectious HIV particle. All right, finally, I just want to wrap up and go through a couple of salient points from my talk. I hope I've shown you some interesting results where our molecule 247 reacts with HIV GAG by this process of acetylation of cysteines and lysines on the GAG. It does acetylate GAG at several cysteines and lysines. We see this both in vitro and in cells. We think we're damaging this large protein in an irreversible manner that, from which the virus can't really recover. There's a really unique pattern, we believe, of this acetylation, and we're trying to link a little bit more closely how this disrupts the eventual downstream processes of viral maturation. I'm going to highlight again, with a little bit of a caveat, that we are not able to generate HIV resistance in cell-based models to this molecule. I think this is just absolutely fascinating, although it's a bit of a double-edged sword in the sense that the inability to generate resistance means we sort of lose out on mechanistic information about how this molecule really is exerting its effect, although we like to approach it from other experiments that we're doing right now. I hope I've shown you some nice data showing that 247 is an effective microbicide against SIV in vivo, and we've shown that it does not cause deurtication, and we are formulating it into different delivery devices. Okay, finally, I'd just like to go through some acknowledgments. I really have to acknowledge the two postdocs in my lab who have really spearheaded a lot of the work that I've shown you today. This work really needs to happen in a collaborative environment, and NIH really provides that kind of collaborative environment, and I've really tried to highlight a lot of the collaborators, and I'm always looking for more collaborators here. I also want to really thank the funding agencies. So I'm supported by the intramural program of NIDDK, and the Office of AIDS Research as well has been very generous and great, and I'm very grateful for allowing us to have the funds to pursue a lot of these studies. Finally, let me just say this is a relatively simple molecule. We produce it in my lab. I'm happy to share as well, and we're always looking for interesting ways to advance this molecule. Thank you.
Thank you, Dan. Our next speaker is Dr. Dima Hammoud. Dima is a tenure-track investigator in the Center of Infectious Disease Imaging, which is a joint initiative between radiology and imaging sciences at the NIH Clinical Center and the NIAID. Dr. Hammoud is a leading expert in diagnostic imaging, PET scanning, neuroradiology, and will speak to us today on molecular imaging to better understand HIV pathophysiology.
Thank you. Thank you very much for the introduction, and thank you for the organizers for inviting me today to talk about molecular imaging and how it can help us better understand the pathophysiology of HIV infection. Just a quick introduction of what molecular imaging is. The official definition is non-invasive, real-time visualization of biological processes at the molecular and cellular levels in vivo. By in vivo, we mean it's in intact living subjects, whether human or animals. In more practical terms, molecular imaging, you could think of it as a hybrid between molecular biology and structural imaging modalities like CT, or structural MRI or X-ray, and the fact that it can provide in vivo data in a non-invasive manner, but at the same time you get molecular cellular information that you do not get from structural imaging modalities. And most importantly, it provides us the opportunity to perform longitudinal observations, which allow us to follow disease progress without disrupting the microenvironment. This is a simplified slide of the major imaging modalities that are generally included under molecular imaging. There are way more derivatives from those. Essentially, you have ultrasound, nuclear medicine techniques, optical imaging, and certain types of MRI. But today I'll be talking, excuse me, mostly about a nuclear medicine imaging technology, which is PET, or positron emission tomography. Positron emission tomography, you're all familiar with it through the use of F18 FDG, which is used a lot in oncology. In which case you inject it into patients and then you obtain a 3D map of glucose metabolism through the body involving the brain as well as peripheral organs. It relies on beta decay of certain isotopes like F18 and C11. There are lots of advantages to PET imaging. And first, that it can be used both in animals and in human imaging, so you can get a very quick translation. It's one of the highest sensitivity imaging modalities available. Very small concentrations of metabolites can be detected with PET imaging, and it has excellent depth penetration. So today I'll be showing you a few examples of how PET imaging can help us understand the pathophysiology of HIV involvement. In this case, I'll be talking about mostly CNS applications. As you know, neuro-HIV is a multifactorial process. However, through the use of molecular imaging, specifically the use of PET, one can parse out those neurotoxic factors and define their contributions to the final result of neuronal injury and death. Some of those processes that can be evaluated with PET imaging include viral protein neurotoxicity, neuroinflammation, which is a big pathophysiology suspected of causing neuro-HIV, lack of adherence to antiretroviral therapy causes and effects, comorbidities, including multiple comorbidities, especially cardiovascular disease, and most importantly, neuronal injury and death, as well as the involvement of specific neurotransmitter systems. Neuroinflammation is a very important pathophysiology that's suspected to cause damage in the setting of HIV. And this is one of the processes that can be quantitatively assessed using PET. Essentially, what happens with neuroinflammation, you have a microglia, which are the resident macrophages of the brain. They get exposed to a noxious stimulus and they get activated. When they get activated, a specific receptor referred to as the translocator protein or TSPO increases its levels of expression. So if you have a ligand that is detectable by PET and that can actually bind to TSPO, you have a method of quantifying how much neuroinflammation there is in the brain. We used one of those ligands, F18-DPA714, which binds to the translocator protein. We used it in SIV-infected animals because we were very interested in detecting the interaction between CSF viral loads and neuroinflammation, especially that recently HIV-positive patients in the early stages of disease have been found to have very high CSF viral loads. So we evaluated our animals at baseline, and then we followed them at multiple time points after inoculation. We were surprised to see that at the early stages of infection, when the CSF viral load was very high, instead of increased binding of DPA714, we actually found decreased binding compared to the baseline. So we concluded that with very high CSF viral load, there does not seem to be neuroinflammation, but rather on the contrary, we are seeing some loss of microglial binding. This pattern, however, reversed when we treated the animals and CSF viral loads went down. At that point, binding actually started to go up, and then it reversed again. And this other animal, again, we treated, CSF viral loads went down, and then the binding went up. In fact, if you look at this last time point here, these are images from the last time point compared to baseline. And you can see that with very high CSF viral load, even quantitatively or qualitatively, you can detect lower binding compared to baseline, suggesting lack of neuroinflammation. On the contrary, when we have moderately elevated CSF viral loads, instead of decreased binding, we have actually increased binding. This is documentation of neuroinflammation compared to baseline. So we concluded also by statistics that microglial activation in SIV encephalitis is inversely correlated to CSF viral load. We confirmed our findings with immunofluorescence. if you look here at the middle row, these are animals with very high CSF viral load, and you can see that there is poor staining of IBA1, which is a microglial stain, compared to a normal animal. At the same time, there's a lot of red staining, which is CC3-PARP or apoptosis staining, suggesting a lot of those cells are actually dying. And that corresponds to low binding that we saw on PET imaging. In moderately elevated CSF viral loads, however, we were able to identify neuroinflammation and some apoptosis, although not as much as with these animals. So we went back to these animals and we wanted to see which cells are actually dying when CSF viral loads are really high. Co-localization with other cellular stains, we found that at very high CSF viral load, actually both microglia and astrocytes seem to be showing apoptotic signs and dying. We concluded that microglia and astrocytes probably get infected early on, and because of very high CSF viral load, a lot of them die, and probably only the surviving ones eventually become reservoirs. More interestingly, we found that neurons are actually dying. So this is neuronal stain, and these are apoptotic stains as well. Excuse me. And the conclusion from this is that there is a certain amount of neuronal death that's happening with very high CSF viral load, and it could potentially account for irreversible neuronal injury that sometimes we refer to as the legacy effect, which occurs early in the infection and persists after antiretroviral therapy initiation. We were also very interested in the effect of antiretroviral therapy interruption. And for this, we used FDG-PET. FDG-PET essentially is a neuronal marker, but if you follow it over time, it could be also an indicator of neuroinflammation. So it could be a neuroinflammation marker, although less specific than DPA714. In this experiment, we evaluated animals that had been treated for 1 to 2 years. We imaged them with FDG-PET at baseline and then at multiple time points after interruption of antiretroviral therapy. And as you can see in the qualitative images, there was actually a significant increase in FDG uptake after 1 month from interrupting treatment, and that persisted up to 6 months afterwards. Those changes correlated with CSF cytokines, suggesting that this process seems to be motivated or associated with neuroinflammation, suggesting that even brief gaps of antiretroviral therapy can potentially cause neuroinflammation and potentially cause neuronal injury. Another process we're very interested in and that we use PET to better understand is depression in the setting of HIV. And depression seems to be a very important cause of antiretroviral therapy interruption. And the recent study showed that HIV-positive patients who are depressed have a striking figure of doubled mortality rates, mostly because they do not follow up and they stop their treatment. So despite the magnitude of the problem, we still don't understand very well why are HIV-positive patients more predisposed to developing depression. So we decided to pursue one of the neurotransmitter systems known to be involved in mood disorders, and that's the serotonergic system. And one way of imaging the serotonergic system that has been used extensively in patients with mood disorders is by looking at the serotonin transporter using C11-DAS, which is a very good and very specific ligand for SERT. So we did this. We used C11-DAS in our animals at inoculation baseline before inoculation, as well as multiple time points over after inoculation, in order to detect those longitudinal changes in SERT expression. And it actually paid off because we were able to identify that in 85% of our animals, there was actually increased binding. These are parametric binding potential maps. There was increased binding in those animals over time, which correlated with duration of infection as well as with CSF cytokines, suggesting some interaction with neuroinflammation. And this is important because this is what's been described in patients with depression and bipolar disease. It seems that increased SERT expression can cause lower serotonin levels in the synapse and potentially cause depression. This is a very interesting study because this is the first time we were able to show in vivo that there is a dysregulated serotonergic system in SIV. And in fact, we're now proceeding with translational imaging with the support of the Office of AIDS Research to further investigate the serotonergic and dopaminergic system in patients who are treated. So all of the previous studies I described tackled mostly the acute aspects of HIV infection, which are easier to implement in animal models. But when it comes to chronic HIV infections and treated patients, a different set of questions arise. And those are best answered in human research, especially in what relates to what we refer to as HIV-associated neurocognitive disorder, which is the fact that chronically infected patients are still struggling with neuropsychiatric problems despite that they are optimally treated and the virus is suppressed. Another question is how much of this neurocognitive dysfunction is due to HIV versus other common comorbidities like cardiovascular disease or co-infections. So we collaborated with Dr. Avindra Nath at NIH, who has a large natural history study recruiting treated HIV-positive patients and controls. There are more than 300 participants now in the study who are all very well characterized neurocognitively and clinically. So in our collaboration, we evaluated 47 HIV-positive patients with brain FDG PET, but because we're interested in comorbidities, we had two groups of controls. We have one group of controls who had comorbidities and another group that were completely healthy. When we looked at the various subcortical regions, the region that stood out was the thalamus. If you look at this graph here, HIV-positive patients showed significantly lower binding or lower uptake of FDG compared to HIV-negative patients and to healthy controls. And in fact, you can see it's qualitatively if you compare the FDG uptake in the thalami to that in the basal ganglia in some of our HIV-positive patients. This thalamic hypometabolism was best predicted by HIV stereostatus, and all this suggested to us there is a persistent viral effect despite optimal treatment. Whether this predates antiretroviral therapy or is still ongoing after antiretroviral therapy is something we need to better assess in longitudinal studies. More interestingly, however, when we looked at the whole brain FDG uptake, we found that HIV-positive patients' uptake was lower than healthy controls, which is kind of expected. You have hypometabolism, but it was not significantly different from patients with comorbidities. And when we looked at various variables that correlated with uptake, the only one that turned out to be positively correlated and significantly correlated was atherosclerotic cardiovascular disease risk scores. So it's a cardiovascular disease marker. And whole brain metabolism turned out to be predicted by cardiovascular disease and not by HIV status. This has significant clinical implications as to the underlying pathophysiology of neuronal dysfunction in HIV, and it really raises the burden on doctors who are following HIV-positive patients that they need to tackle those comorbidities, especially cardiovascular disease, if we want to eventually control the progression of neurocognitive dysfunction in this patient population. So in summary, through the planned and careful use of specific PET ligands, we were able to identify various neurotoxic factors contributing to the neuronal injury and death in HIV-positive patients, thus shedding more light on the pathophysiology of disease and even proposing some changes to the clinical management of patients. At the end, I would like to thank my group as well as all the great collaborators we have at NIH from various institutes, as well as the Office of AIDS Research for supporting us. And I would like to thank you for your attention. Thank you.
Thank you for that talk. Our next speaker is Professor Jeff Crowley. Jeff is the Program Director for the Infectious Disease Initiatives at the O'Neill Institute of the National and Global Health and Law and Distinguished Scholar, the National and Global Health Law and Distinguished Scholar. Jeff's claim to fame is he helped the Office of National AIDS Policy develop the first national HIV/AIDS strategy and was one of the principal architects and is an expert in this area and has remained attached to the field, and his commitment to HIV/AIDS has been spectacular. Jeff is going to talk to us today again about the National HIV/AIDS Strategy, grounding national policy in sound basic sciences. Jeff.
Great. Thank you, Carl. Good morning. It's a pleasure to be here. As Carl said, it feels a bit out of time to be talking about the National AIDS Strategy, which was released in 2010. But I think it still guides where we're trying to go today. But I think the real point is, I know that a lot of scientists often say, and some have said to me, like, we give you the answers. Why is it that it's so hard for politicians or policymakers to do the right thing. And hopefully you'll see a couple things, that your voices and ideas are sort of the basis of a lot of our national policy. But I also think it's a bit more complicated too. You know, we live in a political context. I'd also just make note that Dr. Goodenow wasn't at the NIH when we developed the strategy, but we worked closely with Dr. Weitz-Carver. Of course, Dr. Fauci was a critical partner in this, but you may hear me mention Carl because he was one of my key partners. So I developed a lot of great close colleagues that I'm friends with today. But as we were developing this, there are parts of the strategy or things I'll talk about where I can hear his voice in my head or help me conceptualize certain things. So that's why I might mention him. So I really wanted to focus mostly on like sort of where we are today. So I want to run through some things about like just quickly where are we today. And when I try to capture how do I think of where we are in responding to the domestic HIV crisis today, I'd say it's a time of hope amid some uncertainty. And the question is really, can we maintain the momentum we've generated at a time when our country is so divided? After remaining fairly stable for about 15 years, the recent period has brought some significant progress. I mean, you're going to see Dr. Corey and I had some same slides actually we probably borrowed from Dr. Fauci. I'm not arguing the point. He said we've been stable. I would say we were stable for about 15 years at about 50,000, but recently we've seen about a 20% decline in new infections. And yeah, we'd love it if that were a 50% decline, but an 18% decline is significant progress. And also in our healthcare programs, we're seeing major increases in viral suppression. And yes, we have some major inequities, but we're seeing progress in all areas. So why is that? Why do I think, you know, after being somewhat stagnant, we've started to turn things around? And I always say there's three things. And the first is the National HIV/AIDS Strategy. And again, you know, I was responsible for coordinating its development, but I was not a fan. I thought it was the wrong thing to focus on before we did it. But when we did do it, I think it gave us a focus and gave us created some opportunities to do some politically hard things just by saying this is what we're going to do. But also, I always talk about this, and hopefully you all would agree that I always say that this current period we're in, except for the time in the mid-90s when we got effective HIV treatments, this is the most robust, exciting period of scientific discovery in HIV that we've had. And so what's been interesting to me is not that this new science has sort of changed the course, it's just bolstered the course that the strategy set. And some of this was deliberate, some of it we knew what was in the pipeline or where we thought we were going, but the fact that they work together, and everything is telling us is we've got to get people diagnosed into care sooner. And the third element is the Affordable Care Act. Now, I was on the White House Health team. I don't think I thought or anybody thought that expanded access to healthcare or this healthcare law was the solution to everything. But it was a platform for more progress. So I think it's the fact that they all came together that allowed us to make some of the progress. So the epidemic we have in the United States is the most serious among developed nations, about 1.1 million Americans, about 40,000 new infections per year. I would just put that in context. At the height of the epidemic, we were seeing about 130,000 new infections a year. So that's a major improvement. In our most recent year, we have good data for 2014. We saw about 6,700 deaths directly attributed to HIV. You see roughly, I want to say 15 to 16,000 deaths among people with HIV, all-cause mortality, but a smaller number directly attributed to HIV. As you probably know, the epidemic's highly concentrated in this country among gay and bisexual men, Black Americans, Latinx Americans, Black women, transgender women, and people who inject drugs. Then you go to different communities across the country, you have different highly impacted populations. As was said, the southern U.S. is disproportionately impacted. Half of new infections in the South, where they have 38% of the population. So again, same slide, but again, 51% of new infections are in the South. This, again, same slide, but I think this is stunning. The idea that 46 counties, there's hundreds or thousands of counties in the United States, and 46 counties are responsible for half of new HIV diagnoses. So if we're really trying to have an impact, we need a nationwide response. We need to do things everywhere, but we also probably need to concentrate resources where the transmissions are occurring, and it really suggests a greater focus on these counties. This is a different way of looking at our epidemic, whereas that was focused on geography. This is on risk groups. And if you look at the left, the green side, it shows you that roughly 70% of new diagnoses are among gay and bisexual men. The top 3% are gay and bi men who also inject drugs. 67% are their primary risk factor is engaging in same-sex sexual behavior. 24% are heterosexuals. I would note that These data are from 2016. When we released the strategy, we presented data from 2006. And while the trends aren't different, some things have changed. In terms of absolute numbers, we've known that the rates for Black gay men are higher than virtually any other group. But when we released the strategy, in terms of absolute numbers, it was white gay men, Black women, followed by Latinx gay men. Now we see in terms of absolute numbers, Black gay men way above other groups, but then next is Latinx gay men, white gay men, and then Black women. So all of these are very high-risk groups, but the relationship has changed, and again, it suggests if we're focusing our efforts, which populations may need a little more attention than others. What are we trying to do in HIV? And I think we're trying to get all people diagnosed and suppressed. This shows the continuum. I think a lot of us are sort of sick of seeing it. But the last slide bar on the right, shows that 51% are virally suppressed as of 2015. We need to do better. But I also say when we first started coming up with national estimates, we were at 25% viral suppression. So we are making progress. This slide, so Dr. Goodenow opened up, and one of the things she talked about was U equals U, undetectable equals untransmittable. That's an important thing. It really is very powerful for fighting stigma for a lot of people with HIV. It's changing the dialogue about how people engage with HIV in communities. But it really depends on being undetectable. And, you know, NIH has provided, I'd say, important leadership in a number of ways in sort of validating this, this was a campaign led by the community, but NIH came up and said, you know what, the science is there. Dr. Fauci again has said things, Dr. Goodenow. I actually always, a footnote, whenever I talk about this, I quote Dr. Dieffenbach, and I'm going to just paraphrase it, but it basically says your viral suppression is only as good as your adherence. If you haven't taken your meds in 2 weeks, you're not undetectable. And I think what this chart shows is the durability of viral suppression is too low in all groups. So for all groups, it's below 50%. For men and women, it's below 50%. Varies by race, ethnicity. Black Americans are just above 40%. White Americans are, maybe it's 58%, but below 60%. The highest age group, it's below 55%, but it's lowest for younger people. So there is variation, but we're doing poorly everywhere. So we really, and this is not something to be like depressed about. It's like, this is important information. So it changes how we need to think about our policy responses. So it's not about, did you get to viral suppression once? Do we have the systems in place to support you to stay suppressed over time? So what did we do with the strategy? It was a plan for all stakeholders. We always said it wasn't a plan for the federal government. And sometimes I feel like I repeated this over and over, but I also think people heard it. We developed with broad input. I essentially had a roadshow, went to 14 places across the country, had a community input process. There were community meetings. That's when once we got all that input, we actually published a report saying, this isn't what we think as the federal government, but this is what we heard. Then we had an intensive process with our federal partners, including the NIH, where we sorted through what the community told us, what the evidence was, and came up with a plan. We always said this wasn't intended to be a catalog. It's not saying this is everything we do to fight HIV. These are a small number of high-impact things that we all need to focus on together. 5-year plans, grounded in best scientific evidence. The last thing it says, it had a presidential memorandum. We thought long and hard. I mean, part of the reason why I wasn't a fan of a strategy before I got tasked with developing it is I've been in Washington and I've seen these plans released and forgotten. And so the presidential memorandum and some of the things we did, we're really trying to say, how can we give this some life? And the fact that it was updated, the fact that the Trump administration is talking about an update shows we did something right, because it's carrying forward, which I think is an accomplishment of a lot of people. The default position would have been that it would have been forgotten. Within a year, we had been like, wait, what was that again? I heard something about that. And the fact that we're still using it says something important. Not going to go through all of this. I would say the president, though, gave us 3 goals: come up with a plan to reduce new infections, increase access to care, reduce HIV-related disparities. And we said to achieve these goals, we needed a 4th one, which is achieve a more coordinated national response. Small number of actions. So it's like big ideas about reduce infections. We need to intensify HIV prevention where the epidemic's most concentrated. So let's get away from this idea that we take the money and we spread it uniformly across the country. Focus our resources where it needs to go. Let's stop looking for that magic bullet. We need an effective combination of approaches, evidence-based approaches. Those are the kinds of ideas that sort of underpinned a lot of actions that came out of the strategy. Now, it's built on a foundation of science. We tried to, in the strategy, acknowledge how far science has brought us, but also talk about where we need to go. I'm not going to read this whole quote here, but I seem to remember at the time some advocates were sort of questioning NIH's continued investment in vaccines and microbicides, saying, you know, we've invested so much money, there's been no payoff. So in the strategy, we talk about how important that is and the promise of vaccines and microbicides. Some of this I will say, let me read such part of this because it'll just show you how far we've come since 2010. In addition, an important area to study is the feasibility and effectiveness of using treatment to prevent new infection. Such strategies include PrEP, the use of antiretroviral therapy by high-risk uninfected populations such as HIV-negative individuals and committed relationships with HIV-positive individuals, and potential prevention strategies known as test and treat or test, treat, and link to care. You know, those were new ideas at the time. It was before the IPARC study came out proving that PrEP even worked. We've come a long way since then. Increase access to care. There's talk about the need for more durable treatments, but also we needed more work to understand difference in treatment response between women and men among racial and ethnic minorities. Talked about community viral load. It mentioned NIH's work with test and treat in DC and in the Bronx. So NIH and the role of the investments in scientific discovery were the underpinning of the strategy. This is a chart that was released when the Obama administration updated the strategy in 2015. It was after my time. But let me just run through quickly some of the things in it. Our prevention toolkit is expanded. We now have PrEP, but we have treatment as prevention. We didn't have those when the strategy was released. The Affordable Care Act was enacted, but it hadn't been implemented. We now have 2 new testing recommendations that make it easier to screen for HIV on a population basis. We started using the care continuum. And then the rightmost column, which is probably too small for you to read, it says, research is unlocking new knowledge and tools. Evidence that starting HIV treatment early lowers the risk of developing AIDS or serious illness. Talks about new testing technologies, acute HIV testing, new HIV medications, and the need for continued investigation. This is my last slide. When I talk about where we are, I think it's important that, you know, some of our jobs are to say we want the Congress, the American people to keep investing in the fight. Part of that is we still have 40,000 new infections each year. We have 1.1 million people living with HIV. But I also think part of our job is to get people excited. And not just for where we've come, but where we're going. And I think there's a lot of excitement in the work that you're doing. This is my list of priorities. Hopefully it's a pretty good match with yours, but we need to keep working on the research for a cure. You know, I always hear every time I come to NIH or talk to people from NIH, hear about broadly neutralizing antibodies for prevention and treatment, vaccines for prevention, long-acting agents for treatment and prevention, implementation science to translate research success into population-level impact, and lastly, research on aging with HIV and management of comorbidities. We can do some exciting things in these areas, so we have to keep it going. Thank you.
Thank you, Jeff. Our last speaker, it's always great to have an NIH Institute Director on a panel, so it's my pleasure to welcome Dr. Patricia Flatley Brennan, who's the Director of the National Library of Medicine. And this is her building. So it's great to have, to be here. Dr. Flatley Brennan came to the NIH from the University of Wisconsin-Madison, where she was the Lillian Mulham Bascom Professor at the School of Nursing and College of Engineering. Dr. Flatley Brennan is a pioneer in the development of information systems for patients. The title of her talk today is Data-Driven Cure, How the NLM Inspires HIV Discoveries Through Basic Science.
Good morning, and thank you very much for the opportunity to be here, and welcome to the National Library of Medicine in the room in which you're sitting right now as part of our wonderful operation here. I'm delighted to be able to speak to you about the National Library of Medicine and how data-driven cure can be fostered and inspired by the work of the National Library of Medicine. But I want to take a minute and tell you my personal journey with HIV and AIDS interventions and research. 30 years ago this month, I began a study in Cleveland, Ohio, supported by the National Center for Nursing Research, to use a new idea, computer bulletin boards, to support people living at home with AIDS and HIV. If you remember, in the early days of the epidemic, no one knew much what to do while a person was in the hospital. But when we sent them home, we really didn't know what to do. And nurses were there with those patients and realizing they needed information, they needed peer support, and they needed acknowledgement. Over the next period of 5 years, we learned that providing even horribly primitive computer technology allowed people living with AIDS to interact with each other, to find ideas and thoughts of how to self-manage that professionals never were able to provide, and most importantly, to find a way to live healthfully in spite of being given a death sentence. The important contributions of nursing, of social science research, and of technology to treating the people experiencing the epidemic remains critical today. And that's part of the reason why I'm here at the National Library of Medicine. The National Library of Medicine provides services to accelerate discovery and support information management by individuals. We provide scientific support through our extramural research program, and we provide an intramural space of investigation. I'll be addressing all three of those. But in the services we provide, I'm sure you're familiar with our most widely used service in the world, with PubMed. 5 million people a day are using this service. Some of them are people trying to take care of themselves. In our library services and our library resources, we also provide a trajectory of the epidemic, a trajectory of health through the time in our History of Medicine division. And I share with you here the first approach that we make to collecting the AIDS literature, ensuring the AIDS literature remains accessible, available, and current, and useful to society. Now, increasingly, our PubMed Central, our public access full-text repository, provides information directly to individuals, including over 300,000 HIV and AIDS articles that are cited. We have Bookshelf, which provides full-text access and full-text resource research. In our clinicaltrials.gov repository, we provide access to information to allow individuals to know what clinical trials are available, where are they recruiting, and what might they be providing results about. Clinicaltrials.gov is a 20-year-old operation, but in the last 2 years, we've accelerated the use of clinicaltrials.gov to provide immediate results reporting at the end of 1 year after a trial is finished for all the primary outcomes of a clinical trial. So it's possible to find information directly even before the articles are being written. It's also useful for individuals who are looking to recruit participants in studies. It's also useful to get a landscape of what research activities and research products are under operation and under study. You're probably most familiar with the NCBI's computational and data resources, and I'm going to spend some time talking about this because NCBI, the National Center for Biotechnology Information, actually grew up along with the epidemic. The NCBI was formed in the mid-1980s with the recognition that genomic inquiry, the genome, was going to become the core basis of our understanding of health. And in order to understand health, we had to be able to curate, hold repositories, and improve the best practices of use. The National Center for Biotechnology Information is a critical resource for AIDS research. They importantly do three key characteristics. First, we provide repositories that store information, databases of HIV and AIDS variants, including over 700,000 HIV-1 sequences. Secondly, we provide tools to support the HIV and AIDS research, most importantly, our BLAST tool, the BLAST sequence similarity tool that examines the identification and alignment of different sequences. We allow for global alignment for rapid identification of new mutations in HIV sequences. And we've increasingly in the last few years been building a greater set of tools for decision-making, tools for visualization, and tools for searching in sequence. Finally, through the NCBI, we provide information about HIV and AIDS clinical trials. And these clinical trial listings that I just mentioned to you are not simply an enumeration of trials, but also are a pathway into looking at the literature, reporting that information to the gene sequences that are involved in those studies. We've made a particular effort here to curate protein structures. And NCBI's collection has almost 1,600 protein structures. Here you see the crystal structure of the HIV GP120 protein with a bound antibody. Our attempt to create these structures is not only to hold them in a repository, but also to make them accessible and available for inquiry, for examination by investigators. Perhaps our most important resource is the HIV Human Interaction Database, over 2,500 manually curated interactions. This database includes all the known interactions of HIV in the human host, including human genes that have been reported to affect the viral replications and infectivity, the interactions between HIV proteins with host cell proteins, other HIV-1 proteins, or proteins from disease organisms associated with HIV. Documenting this is essential because it allows us to understand the process of HIV replication and pathogens and provides a foundation for researchers to develop safe and effective therapeutic and prevention strategies to combat AIDS. We provide the in silico operations to inspire new investigations, to inspire new approaches to study. In conjunction with GeneBank and the HIV-1 human interaction database curation efforts, the NCBI has developed some very specific tools for curation and analyzing HIV and other retroviral sequences. For those of you less involved in the curation process, let me explain that each time a sequence is deposited at NCBI, it goes through a many-step series of human assessment to ensure the integrity, the appropriate identification and documentation, much of this involving interactions with investigators. Once a sequence has been properly curated through the NCBI, it is available and made public for the use of the entire scientific community. We do follow the NIH genome data sharing policy in all of these activities. That in turn leads to sometimes a little bit of a more cumbersome interface to access some of our genome sequences because of the need to protect the conditions under which the data were acquired. Nonetheless, once we were able to bring a sequence in, we were able to apply genotyping tools using the BLAST sequence similarity algorithms to identify the genotype of a query sequence. We're then able to allow researchers to quickly characterize novel HIV genome and compare it to previously established HIV types. There is a global alignment tool for rapid identification of mutations in the sequence and a collection of 66 retroviral genome maps, which provide a reference resource for these. So we are building a library now not with books and articles, but with genomes, genome sequences, and the tools that make this useful to our society. The tools that you may be familiar with are the structure that we've put in place for the reference sequence, including the protein accession numbers. Right now, we've accelerated the intake process such that it's possible to get an accession number within 24 hours. So we're able to move the pipeline more quickly. Our entrees at genome ID numbers are assigned once the curation process is complete. We're able to identify and link to known amino acids and provide brief human-driven descriptions of the interactions. We have over 300 curators that assist in the documentation and curation of our genomic structures, making sure that the scientific content needed to evaluate and describe the information is available at the point of need. We also, though, link this information to the literature and using keyword support can make connections to the PubMed resource, building the background of this, building the platform that allows us to connect from a genome to an article to a clinical recommendation requires conceptualizing of a data architecture that is leading the NIH in its building of clinical information resources. Our resources are also useful in surveillance of HIV mutations in individuals. We do understand that HIV has a very high mutation rate, and the sequence is needed to determine for evaluation and comparison. Our process links together three key resources, PubMed on the left, GeneBank on the right, two of which are held here in within the NLM, and then the Stanford Drug Resistance Database. This process allows us to understand the strategy of treatment and to provide combination effective antiretroviral therapies effective against the resistance in a timely enough fashion. It essentially is bringing together patient virus and population strategies to inform the treatment. Our population detections and surveillance tools now, it uses a BLAST sequence to identify the genotype or subtype of the HIV sequence, and you see one of our graphics displayed here. We're increasingly looking at making more effective visualization tools for human use, but also recognizing that increasingly the interaction with our databases and data repositories are driven by machine interaction, machine learning, and machine algorithms. So we recognize that our consumer is increasingly the computer. Nonetheless, population surveillance tools allow us to be able to take geolocated information, our own genotyping information, and the drug resistance, and understand patterns that occur through the country. This is still requiring an intensive amount of human engagement to understand how we bring these three elements together, and will not, in the short term, be done only by machines. The vaccine and targeted discovery process is supported also by the NIH. The National Center for Biotechnology Information brings together information that allows for testing and to identify novel drug receptor sites and novel enzyme interactions that can be determined here. We are working specifically, though, to increase our understanding of clinical information, aligning that with the biological information that comes through. So I'm now going to talk with you a little bit about another part of the NIH National Library of Medicine resource system, and that's what the work that is going on in the Lister Hill Center Computational Clinical Support Services. Lister Hill Center has been working to enhance the identification of research common data elements in HIV and AIDS using data science methods. So within the clinical trials documentation, where we are emphasizing not only the declaration of a trial, but the sharing of data, we have the capability of identifying the specific clinical variables that are used during research studies by appending to a clinical trial record a series of individual participant data sets, as you notice on the right, as well as the study protocol, study forms, and manual procedures. Common data elements represent a commitment by the NIH to ensure that the variables used in human studies can be integrated across multiple human studies. It's an attempt to provide common language, common terminology to ensure that patient outcomes in these various studies are detectable and traceable in a way that can be generalized across several studies. A key component of the study of this process now is a collaboration we have with the National Institutes of Minority Health and Disparities, where we are looking to establish common data elements that vary specifically by various socioeconomic or identity and ethnicity classes, so that we now move away from smoothing our interpretation of clinical phenomena over large groups of patients to being able to detect specific clinical phenomena. Another part of work that's going on here in the National Library of Medicine may surprise you, and that is the use of chest x-rays and automatic detection of pulmonary disease in the treatment of people with AIDS. The National Library of Medicine applies machine learning to images to be able to detect variations and perturbations, particularly that identify individuals who are developing complications, tuberculosis, and other kinds of pulmonary disease. Excuse me. The work that is going on has led to the development of an in-field interpretation system, so that as a patient has an X-ray completed, the X-ray is evaluated, transmitted to a secure system, and a machine learning algorithm applied to determine in the moment, is this person likely to be demonstrating signs of tuberculosis? This has allowed us to accelerate the treatment of people who have secondary consequences along with HIV diseases, to allow for one visit to the clinic as opposed to multiple visits. In low-resourced area, in highly remote areas, being able to apply machine learning to clinical images in the moment is going to accelerate our ability to engage people in treatment as early as possible. Our resources are now being expanded to look at TB within intramural experiences here in the clinical center, and we expect to see this area growing. This operation, by the way, is not in full clinical practice now, but is showing great promises. I want to close by reminding you that the National Library of Medicine serves as a place to collect and perpetually store information that's relevant to the health of society. Much of that information has to do with accelerating basic discoveries and providing the platforms and tools that NCBI provides to accelerate how our scientific researchers are thinking. But we also provide information that reaches out to the public. So please, as you finish your visit here this morning, take a few minutes to look at our banner displays out in the hall. We have created banner displays to expose and make people familiar with the many holdings of our History of Medicine division. This allows us to send these banner displays to public libraries, to high schools, to community centers around the country, increasing the awareness of the history, the challenges, and the hope for the treatment of AIDS and the prevention of future consequences. Thank you very much for allowing me to share my perspectives with you.
That was quite a set of talks. I want to, again, please join me in thanking the panel for their presentations. So now we're to the open mic part of the morning, and I would like to just start a moment and make an observation that the first talk was Dr. Corey talking about a vaccine that is in 705 that uses something called a mosaic, and that was built upon the platform that was just discussed in terms of what was built at Los Alamos, handled here. So that is, from beginning to end, a perfect example of how the basic science of sequencing virus after virus after virus, collecting, curating, making sure that we had clean, clear viral sequences, clever people at Los Alamos working with the database, designing immunogens that are now going into thousands of people around the world. So as we think about where we are in HIV/AIDS today, that kind of linkage is really impressive, that it occurred centered within the NIH, but it has such a global impact. So I'll open the floor to questions in a minute, but what I'd like to do is first pose a question to each member of the panel, and it's a two-part question. What do you think is the most important finding that we've had in HIV so the first one's a lollipop question, as you can tell, over the past 30 years, and what is the remaining biggest challenge you face in your work that you think basic science needs to help you with? I'll pick on you first, my friend.
Well, I guess I'd probably just say isolating the virus and identifying it as the causative agent. And I think at the moment, the biggest sort of challenge is to sort of how to use reverse vaccinology. We know where the neutralizing antibodies bind, and we know sort of where they are on their epitopes, but designing an immunogen that actually keeps that in a stable way and actually in a multiplier way, because most of our effective vaccines have been either synthetic self-assembling proteins that give multiple copies, 50 or 60 copies of a very stable capsid structure. And so that kind of stability for the trimer has yet to be achieved. So I think we'll need to sort of understand the spacing, the baldness, and how to get the multiplier effect in order to make that kind of immunogen.
Thanks, Carl. First of all, let me just say that I've really enjoyed the talks, and they're superb and very thought-provoking. And so I've got to thank you and Maureen for organizing this workshop on this very important day, which is the World AIDS Day. So when I look at the central nervous system, I have to say that the biggest advancement for neurovirologists was that, before AIDS came around, we always thought that the way you cause destruction in the brain is the virus infects the neuron, it kills the neuron, that causes symptoms. And that's true for rabies virus, for polio, for herpes viruses. But when HIV came around, that's what we thought would happen too. We looked at the brain and there was no HIV in the neurons. Yet all these patients were getting demented. The neurons were dying and there's no virus in the neurons. To our surprise, what we found was the virus was sitting in these glial cells. So we thought that is rather strange, but that advanced our understanding of a lot of other neurobiological diseases, because we found that there is actually indirect damage in all the other viral infections. So the number of infected cells are actually very few in the brain, but the amount of damage that occurs to these cascade events is actually huge. So we thought somewhat naively that now you have an infected cell and you have the damaged cell, which is the neuron. All you have to do is put some kind of a barrier around the infected cell, and if you could put a condom around it, maybe you'll prevent the damage onto the neurons. So we tried everything we could.
and calcium antagonists and glutamate antagonists and all kinds of neuroprotective drugs, antioxidants, they all failed. All right, nothing really helped us. So the biggest challenge still remains in our mind is really what to do. How do you prevent indirect neuronal damage? And as we're thinking about cure, how can we actually excise this virus out of a brain tissue without causing collateral damage? Everything we've thought about how to do it, what you have to worry about is that you're going to cause damage to other things. So I think those challenges still remain for us.
I would argue that that applies to the immune system as well. We have a fundamental lack of understanding how to eliminate HIV globally in the body. Daniel.
Thank you so much. And I guess from my perspective, sort of being more in drug development, I think one of the most remarkable realizations is just the resistance, which really complicates any kind of drug development program to treat HIV infection. The fact that the virus mutates, and the paradigm is that every medicine that you throw at HIV virus will eventually result in some kind of resistance development, I think is one of the most fascinating things, but also perhaps maybe one of the most challenging things in terms of developing medicines to sort of combat this. And I think I'm awestruck, even as more and more potent medicines are developed to treat HIV, only a slight change in the genetic makeup of the virus to give a slight amount of resistance is enough to give the virus an escape path to then cause additional damage. And it's amazing because the frustrating thing, I think, also results in the fact that this really makes the treatment so important, because if any individual sort of falls back on treatment, even to a slight amount, that opens up a window for resistance to start developing. As far as challenges, I mean, I guess for myself, I think the most important sort of dream for me is to see our molecules enter into humans, and finding a path for that to happen. I keep exploring from different fronts, but it's sort of a challenging path to sort of navigate, going from what's called bench to bedside. And although we talk about it happening, the path is never sort of a straight line. It's sort of forward and back and forth to try to see what really is the best way to advance this molecule. And I'm still sort of searching for that path. Propping it up with basic science, but also trying to navigate the translational framework to translate it forward. Thank you.
Thank you again for the invite and for this great opportunity. So I'm an imager, so I guess the most impressive thing to me has been the development of antiretroviral therapy that, even if it didn't achieve cure, has actually turned this disease from a deadly disease to a chronic disease. But I also agree with Avi. One of the greatest challenges at this point is, despite the fact that we are treating 50% of the patients who are infected with HIV, and we're achieving viral suppression, there's still a lot of neurocognitive dysfunction that's happening in those patients. There has been a lot of work that's been done recently to better understand what's going on, what's causing this, and what's the pathophysiology underlying it. And there's a ton of work that's being done. But from my perspective as a radiologist and as an imager, as a molecular imaging person as well, I think one of the greatest challenges is to be able to establish this connection between the two fields. So there's a field of HIV researchers who know a lot of the disease, know a lot of the pathophysiology, and then we have the other group who are the imagers who know a lot about the imaging. The interaction between the two groups is still lacking. And I was very fortunate because I was working with Dr. Nath since the beginning. I also was working with Dr. Lane, who was very interested in HIV and imaging applications in HIV. But I believe that molecular imaging modalities have great potential to not only understand the pathophysiology, but eventually assessing the effects of vaccines, for example, on disease development. If we manage to follow the viral infection progression, like with antibodies or nanobodies or various molecules that could detect the reservoirs, for example, one could achieve a lot in a non-invasive manner and in a longitudinal manner to follow disease progress. And really all we need to do is just to sit together and talk about it and ask the questions from both perspectives and then come to a consensus on how we can both help each other. I think that's my perspective, which is biased, I guess.
So I think I'm the most removed from basic science, but I guess I would say that the biggest advance to me is just the array of effective treatments. But the way I look at the world, I think, the impact we've had has been remarkable for individuals, but it's really how do we get to population level impacts with everybody? And, there are these huge structural barriers that we all know about, poverty, inequality, like there's just so many. I don't know that that's NIH's responsibility to address those. But I guess when I think about research that really excites me, some of it's around long-acting treatment and prevention. I don't think it will solve every problem, but if we're moving from expecting perfection as far as pill taking daily to doing something a couple times a year, maybe we can make more progress at population level impacts. So that's just one thing I would point to.
Thank you very much. People are alive, and that's the most amazing thing to me. I met a 70-year-old person with HIV. That was inconceivable in the beginning of the epidemic. So yes, the greatest thing that's happened is treatments have some effect in some populations, and people are alive. The challenge is that a 70-year-old person with HIV needs daily reminders to stay on medication, needs support to deal with secondary challenges. And so there's a clinical cascade that has to be engaged, just as Dima mentioned about the link between imaging and basic science researchers. We need that link to expand into clinicians so that it is not simply the treatment that's provided, but the strategies to promote treatment, to maintain engagement with therapies, to anticipate what could be happening with the person. And in our case here, particularly in the library, it's recognizing that information not read is not helpful. So we need innovative ways to get information, not only about the latest discovery, but how things fit together, how your life changes with these, into the hands of individuals. So we're looking to innovations in visualization, in virtual reality, and ways that will engage patients in their lives better.
I think that's a really excellent point. And I think one of the great strengths of the research that NIH supports is it's not just basic biomedical science. And both you and Jeff just talked about the need for behavioral science, because ultimately, as we think about adherence, it is a personal battle on a daily basis. What can we do to understand the motivations and the structural barriers to adherence? And where appropriate, what is the research that NIH can support to help drive that forward? It's absolutely true that long-acting will be a tool, but like we say, pills don't take themselves. Long-acting agents also don't take themselves, and I think that will remain a challenge as we go forward. I'm happy to take questions from the audience, so if you have one, there are microphones around. You can raise your hand. Additionally, were we going to get questions online, Yvette, or did none come in? So we have a question in the back.
Thank you very much. This was very, very valuable. I actually have a question to Mr. Jeffrey. We learned from your presentation that it's very important to diagnose the HIV patients earlier on. So what are the steps? And I don't know if this is an NIH thing, but what are we doing in terms of research to get better at reaching more people faster and getting cures to them sooner than later?
Right.
And I don't know if this is the right question for this platform.
No, I think it is the right question. And again, I said when we were developing the strategy, some of this I can think to conversations Carl and I have had. First of all, I would say, there's a lot of metrics where when we compare ourselves to developed nations, we're doing poorly. This is one metric we actually do well. Like our diagnosis rate is relatively good. Previously, though, we were good about paying for, like, if you went to your doctor and said you engaged in a behavior, thought you were at risk, they could pay for a test for you. We had a weak spot as far as paying for just population-based screening. Some policy changes have happened both a few years ago and also last week to really facilitate the idea we should just be routinely screening people for HIV when they ever come in for clinical care. That's part of it. I think CDC has also done work with what they call their expanded testing initiative, showing that by really focusing resources on some of these highly impacted communities, that you can increase diagnosis rates. But when we released this strategy in 2010, the HHS guidelines are in some ways, I think it was the NIH guidelines, although Carl always corrects me. They didn't recommend starting treatment right away for all people. It's really still based on CD4. And we had a conversation. He's like, I think there's some holdouts on the panel. Didn't think we would get there, but they got there. And I think for a while there was some consternation, but I think now everybody agrees we should treat people, or at least offer treatment right away. So that's been a change. But now I think what we need to do is build the system. So if we're doing good on screening people, our weak spot is the linkage, especially if you're screening people in community-based settings. How do you make that handoff to a clinical care site better? I think, I'll stop talking after this, but on all of these issues, I feel like I can point to some bright spots around the country. Our challenge is taking these bright spots, model practices, and making it standard practice everywhere.
Great, thank you.
Paul.
Thank you. Thank you very much, and thank you for all of the excellent talks and presentations. I have a comment first and then my question. Building on what Dr. Dieffenbach just said, and essentially riffing on the reporting out of resistance currently with frontline meds, currently at 10% in a number of countries, reminds us that we will not necessarily treat our way out of this. We do have stigma dynamics, we have adherence dynamics, etc. And as Dr. Dieffenbach just reminded us, there's important basic behavioral and social science oriented research that is going on at the NIH and needs to continue. My question is for Dr. Hammoud. And with your depression findings that you reported, did the investigators differentiate the depression that was being treated with antidepressants and which type, whether there were serotonergic oriented medications versus untreated depression?
Well, actually, the work that we've done and that I showed was only in SIV-infected animals. So at this point, we were not able to perform behavioral testing, mostly because there were lots of confounding factors, like the patients being sick from the infection. The next step, though, is in collaboration with Avi, we're actually now evaluating patients who are treated with and without depression. We had previously shown in a study that we did at Hopkins a few years ago, we showed that depressed HIV positive patients had higher SERT binding than non-depressed HIV positive patients. But those were very heterogeneous patient populations. Some were treated, some were not. Now what we really want to see is in a fully treated patient population, what are the findings? Can we reproduce the animal research findings? And then we're going to correlate with depressive symptomatology. We cannot really image patients who are on antidepressants, mostly because the majority of antidepressants are SSRIs, and those block the same target that we image. So they have to be untreated patients, which generally means they have to be, if they are depressed, they have lower levels of depression. But we'll see how those findings come through. We already started imaging patients.
Thank you.
Peter.
Thanks, Carl. I'm Peter Kilmarks from the Fogarty International Center. I just want to start by really thanking and congratulating Maureen and Carl, and this has been an incredible panel and really diverse set of information, and I think a wonderful way to commemorate World AIDS Day. I have a question for Larry and then a question stimulated by Avi's presentation, although I'm not sure if he'll be the best person to answer it. But for Larry, you described this remarkable, now 3 large trials in the field. And just with my Fogarty hat on, wanted to ask about capacity. And I've heard Glenda Gray talk about challenges in finding enough trial sites, or with really what's in the pipeline, the challenge of developing enough sites even beyond South Africa, throughout Southern Africa, where there's a high enough incidence, and what your perspectives are on where are we in terms of capacity and what is needed from Fogarty, for example, to help to meet that.
Well, keep on going and expand. Actually, yeah, the HVTN now we have 52 clinical trial sites. And I would say that that the rate limiter at the moment is actually, I think, investigative human clinical trial. It's not the infrastructure, it's the people, especially the leadership in the physician scientist. That career path in Africa is really not well established, especially for males, male Africans. And not that that gender makes a difference. It's just sort of the issue of getting enough capacity. So, our studies now, we're in instead of 9 of the countries in sub-Saharan Africa, certainly we have over 24 clinical trial sites in South Africa alone. But it really is the human capacity issues of getting research leaders at all levels, physician levels, nursing levels, and public health levels to do that. And it takes time. It is, we have been working for 10 years to build our infrastructure for where we are. NIH has been critical. We've had a wonderful partner in the Gates Foundation in helping us, and are very thankful for that. Fogarty program, Glenda's Fogarty Fellow, Linda Gale Becker. I mean, if you go through the list of who are key people, they have come through the Fogarty. And so we are quite appreciative of what the Fogarty does and its long-lasting effects.
You had a follow-up question for Avi?
Thanks, Larry. And so my second question at the end of Avi's presentation talked about using CRISPR-Cas to eradicate the reservoir in mice, and kind of stopped there. And that's sounded very intriguing. And I don't know if Avi or others on the panel or in the room can talk more about the promise of that.
Thanks very much, Peter. I thought you were going to ask me something about international stuff. That's one thing's going to be hard to export internationally. Yes, so there is some exciting work that comes out of originally from Camille Khalili's group at Temple University. And so what they did was they designed these guide RNAs against a portion of the LTR and another one sitting in GAG. And so they were able to excise that region there. And then they used another set that goes from LTR to LTR, actually, so they can pull the whole thing out. And so initially they showed really nice data in vitro. Then they published another paper in Science Reports showing that they could take this transgenic rat model and then excise it there too, actually, and surprisingly within the brain as well. Okay. Now, what is not entirely clear is what percentage of cells they are getting. They claim that, with subsequent rounds of it, they can get nearly everything cleared out, which is incredible, because in a transgenic mouse model and a rat model, you've got it almost in every cell. And so, but now they've got some private funding, and along with a group in Nebraska with Howard Gendelman, they formed a little startup, I think. And they've got other ways of delivering these things with nanoparticles, and they're going to some protein ribosome mediated delivery systems. And so it actually, the data still looks quite interesting. And so eventually I'm sure that they're going to try some pilot study in humans. So I think there's a reason for optimism with that technology.
I would like to add that they're using the CRISPR-Cas9 or other methods to create the CCR5 negative cell types. Has moved into clinical trials. Carl June and his company and Paula Cannon through the California system has moved a number of these into people over the years. So I think the strategy of creating resistant cells by a variety of mechanisms, David Baltimore had a series of strategies for that as well as tackling the virus, will remain an open area of research for a while. I think the biggest challenge with all of these remains the off-target effects. And I think that that's one of the things that the Temple group really needs to worry about. I think that when you have something as specific as an LTR activity, and given the fact that there are human retroviruses within our genome, you run the risk of all kinds of things, because there are actually human retroviruses that exist in our genome that are critical for life. They've been adapted. So I just think we have to be very careful going forward. And we will not talk about China. So, Geraldina.
Just a short question. Thank you for the talks. They were wonderful. But this one, I have a very short, simple question to Dr. Appella. So is your compound strictly antiviral against HIV, or are there other viruses that it could possibly be useful?
Yeah, as far as we've screened a few other viruses, never really saw any kind of reliable antiviral activity. And again, we've been trying to drill down to why. Part of it, I do think, stems from the unique structure of GAG that HIV has, and even more specifically down to the nucleocapsid protein, which we're sort of linking a little bit more strongly now as to perhaps being the real sort of initial attack site. And HIV is rather unique in having the highly conserved nucleocapsid protein that it does with two zinc-structured regions that are essential for binding to RNA. And that region also is very highly conserved across HIV viruses, which we're also sort of postulating might be one reason why HIV, other flavors of HIV can't really find an escape route from that molecule. So yeah, we have hope that we can kind of keep pushing this forward.
Can I just follow up on that question? You mentioned the retroviral zinc fingers. Does this, what is your level of cross acetylation of other zinc fingers that exist within the human cell? Because there's a whole series of proteins that are zinc fingers.
Yeah, no, of course, that's an absolutely great question. And the story always gets more complicated. So I've shown you a bit of a snapshot of the mechanism as we understand it now. And so, yes, if you certainly take this molecule as is and expose it in vitro to other zinc fingers, oh yeah, you'll get reactivity for sure. However, the mechanism of what we're sort of looking at is a lot more complicated with this particular molecule, in the sense that if we were sort of non-specifically targeting just endogenous zinc fingers with our molecule, I'm sure we would have seen toxicity come up, which we really haven't. So there's another layer to this also, in the fact that I would say that most endogenous zinc fingers crucial for the cells are nicely locked up in already important cellular interactions. Not as accessible as you might think, perhaps, to our little molecule. However, in HIV's effort to really push forward in replication and produce an enormous amount of GAG protein, I think that creates a very kind of unique viral environment where our molecule is able to sort of wiggle in and take advantage of sort of a very unnatural environment in the cell for our molecule to sort of attack it. Now, I want to say there's one other layer to the mechanism that really requires a lot more time for me to explain, and I'd be happy to do so. But the active form that I've showed you of our molecule, it doesn't exist in a very large amount within the cell. There's a whole other interesting pathway we're trying to uncover in which our molecule, that acetyl group that I showed you, actually doesn't come from our molecule. It comes from acetyl-CoA. And that's actually kind of another interesting facet in the fact that while I show it as an activated form of our molecule, really what our molecule does is promote an acetyl reshuffling from acetyl-CoA in the cell to different targets, in particular GAG. And that's really very strange in a lot of ways. And it's something, again, we still are trying to wrap our heads around more specifically. But I would say also that it's really this just unique environment in which GAG has to be produced in large quantity and so quickly, and this drive to assembly. And I think it's really in that sort of microenvironment that our molecule becomes active and is allowed to then acetylate GAG in those unique patterns that allows it to actually exert its antiviral effect. I'd be happy to talk more about all this, as you wish.
It's really interesting if you think about what we heard from the National Library of Medicine and the protein interaction database, and the fact that there are thousands of proteins that are involved in HIV interaction. And we think about what we consider the important targets for HIV, and we have the classic big ones of integrase, protease, reverse transcriptase, and entry. But we also have the host factors that we know are essential and play a big role, like apobex and others. But there are other host factors and other pathways that have been mapped, like acetylation or like other forms of host modification that occur in the cell that we don't truly understand how that works. And so there's a whole other layer of basic science that I know that the basic science program of the Division of AIDS would be very interested in grants in this area to really dig in to, are there other mechanisms that we really truly don't understand, such as acetylation, that could be playing a role, that could become targets, but ultimately will tell us about just knowledge about what goes on in the human cell that is essential for life.
So, Carl, can I add a couple of comments to that? I think that's a very, very fascinating area. And, you triggered discussion around endogenous retroviruses. And so one of the things about endogenous retroviruses is that, they are very active early in embryogenesis, but then after that, they shut down. And they may get reactivated in cancer and, in other diseases. So there's a whole viral regulation mechanism that already exists for these endogenous retroviruses. And if you were to be able to understand that, that could shed important light on how to really control HIV, because we're so good at controlling these endogenous retroviruses in adults, and we could probably adapt that same system towards HIV. The other thing is that, you talked about, the overlap between HIV and endogenous retroviruses, and that brings up a very interesting possibility that, so I was going to ask Dr. Corey that as we think about vaccines against HIV, we also know that there are recombinations that occur between HIV and the endogenous retroviruses. Is there a possibility of an unintended consequence whereby we get really good at controlling HIV and we may now drive the evolution of these recombinant forms and now have a new virus that might emerge instead?
Well, the vector systems that we've been used have been replication incompetent vector systems, so we've sort of stayed away from that issue. I guess everything is sort of possible, but that awareness has sort of driven sort of the safety part of replication deficient approaches. I mean, we're just starting to put replication competent viruses in, CMV, some VSVs. We sort of stay away from the lentiviral approaches.
No, I wasn't thinking in terms of vector, but I'm thinking about the HIV that you're trying to get rid of with the vaccine. That itself may integrate into the human genome and form.
Yeah. So I think the challenge there, Avi, is it would have to actually be assembled, make it into the blood and ultimately into transmission. I think it's fascinating, though, that there are groups, including Doug Nixon's, who was formerly in Washington, D.C. and has now moved north, has looked at the immune response against retroviruses and shown a degree of correlation between protection from disease and an immune response against retroviruses that are part of the human genome. And that is an active area of work in cure that I think is really important for us to consider. Yes?
I'm going to give you an opportunity to ask me for some work in this area, because when I listen to what you're describing, what I hear is a tremendous need for effective experimental planning, for effective recording of intermediate steps during the discovery process, and for understanding the context of as well as the individual process. And the library is well equipped for the one-to-one mapping. So we can give you an article for one question, and maybe we can cluster articles together. But we recognize that with emerging data science techniques, partnering with domain scientists, we might be able to accelerate experimental discovery better. So if there's ideas with my NIH colleagues or nationally where the NLM investment in analytics, research planning, and also data discovery, we would be very interested in some partnerships and investigating in that area.
I think it'd be very worthwhile getting a group together to talk about pathways and also different mechanisms of whether we're talking about acetylation or hyphosination or other known activities that we have a glimmer of what's going on, but we just don't really get exactly what is the exact pathway. I mean, some of it's well mapped out, but I think there is an opportunity here to explore more in some very interesting ways. I think it is important that we move from the single one-on-one interaction to domains at this point. If there are no further, we have one more question from the back.
Good morning. I gotta lower this. I thought I sat here just so that I can reach it. Obviously, I still can't. Okay. Thank you all for coming and for your wonderful, informative presentation. I have a question to Mr. Crowley. Thank you so much for sticking in for that resistance, and then you stayed in and go for it, and now you're updating the 10-year plan, right? I could be wrong. I am a slow reader, but I did not see in your update strategy a bullet with regard to behavioral, because in my opinion, each one of us, it all boils down to behavior. Am I going to engage into X because of my behavior? Am I going to engage in participating to the rest of the cascading effects of research and that because of my behavior? Am I going to read what NLM is putting out there for information because of my behavior? So I just would like to know if your office would consider in updating your strategy to put in some kind of bullet in there that really behavioral, a bullet for behavior for people to understand. How do we understand people to engage, participate, and inform, educate? So that's one.
I'm not going to force Jeff to answer that question because he's not involved in the national AIDS strategy anymore. But I because the O'Neill Institute is part of Georgetown. The federal system is busy revising the national AIDS strategy. I would say behavior doesn't need a specific bullet because it is embedded in everything that we talk about, from the behavior of testing and how you engage in testing to care delivery, where it is all about the behavior and the personal interactions. So I would say to pull it out would diminish its value across the strategy.
I see. And the second question is, can you give us an update on PEPFAR? Because it's interesting to me that South Africa still has 53% of HIV-infected population.
So PEPFAR is now 15 years old. It was the highlight of Mike Pompeo's speech and what the Vice President of the United States talked about yesterday. It is going to be reauthorized, which is a major step forward. And it is the major success, one of the major successes that we have had as a federal government in terms of combating the international epidemic. I think that we can learn some lessons domestically about how to do better, and I think that continues to be a challenge. And in many ways, the National HIV/AIDS Strategy tries to learn from what PEPFAR has done to help improve care delivery in the United States. With that, I'd like to call on Dr. Goodenow to wrap us up.
Wow, what a morning. Thank you so much. I want to thank our wonderful panel of speakers. Carl is our moderator. I want to thank the National Library of Medicine for this wonderful facility. I'd like to thank all of you in the room and the more than 125 people who have been listening in on our live Twitter stream. Yes, thank you. I also want to thank Yvette Edgehill-Spano from the Office of AIDS Research and her amazing team for the outstanding planning and execution implementation. It's been a really fantastic event. We've heard some really exciting talks that really cover the spectrum of the HIV research priorities at the NIH. We heard a fantastic prevention strategies. We heard about persistence, reservoir, and cure. We heard about next generation treatments. We heard about pathophysiology and novel ways using imaging to look at co-infections and comorbidities associated with HIV. We heard about the big data and all of the fantastic resources that the National Library of Medicine have and how we can continue to develop using those to sort of complete and go around in the circle of making new discoveries and designing new experiments. And then finally, we heard about how research really does have an impact on policy. I was thinking when Jeff was talking, we talk about how every vote counts, and there was just some anecdotes this morning, I think, on NPR about how one vote in so many cases across the country made a difference this year. And I think that's really important for the researchers, for all of us in the field to understand how important what we do is for translation into policy that really puts into practice what's been discovered. And PEPFAR was mentioned, and I think one of the ways I think about this is when you look at that pipeline that I showed you that starts as basic and goes to public health, PEPFAR is an example of our ultimate consumer and the way we really want our discoveries at the very basic end to have an impact on public health. We also heard about the National HIV/AIDS strategy that's in place till 2020. And that strategy has terrific synergy with the NIH HIV strategic plan for HIV research, as well as the research priorities. And all of these are in place till 2020. And so there's a lot of activities going on in different spheres to set up what the plan is going to be from multiple parameters from 2020 to 2030. I think what we heard today is an amazing pipeline, lots of new excitement and new discoveries, and a lot of work to do in the field. We talked about the global epidemic to some extent, but focusing in the United States today, don't forget that there are 40,000 new infections a year, which is more than 100 per day. So we have a lot of work to do internationally and domestically. I want to thank you all for celebrating World AIDS Day 2018 with us here, and look forward to a very productive year. And thank you again to our panelists and to all of you for participating.